I want to welcome you to RPB's Lunch and Learn Eye on Sjogren's disease, which you may know as Sjogren's syndrome. I am Brian Hoffland, President of Research to Prevent Blindness. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. RPB was founded in 1960, and over our 63-year history, we have awarded more than $400 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. In today's educational session, you will hear from two RPB-supported talented scientists about Sjogren's disease and its causes, impacts, and cutting-edge research. One of our speakers, Dr. Esen Akpek, received an RPB Special Scholar Award, and the other speaker, Dr. Cynthia DePaiva, has a current RPB Stein Innovation Award. Thank you to all the generous supporters of RPB who make our work possible. I'd especially like to thank Dompe for their support of today's educational event. The importance of supporting medical research has never been so apparent. We thank you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. If you have not yet supported RPB, please consider doing so to help make sure that we are able to continue with these breakthroughs. Getting back to today's session, let me introduce our moderator and friend, Georgia Posadas, PharmD, RPH. She joined Dompe in 2018 to build the Medical and Clinical Affairs organization. She is currently the Senior Vice President of Medical and Clinical Affairs, where she oversees the field medical team, medical information, and communications and operations. She also oversees new indications within neurotrophins and post-marketing clinical development and clinical trial operations. Our moderator will introduce our two speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes, and I will serve as timekeeper. I will give each a warning at the 18-minute mark. We want to make sure that we have time at the end for a robust Q&A. Georgia, take it away.
Wonderful. Thank you so much. Hello, everyone. It's a pleasure to be here. And thank you to Dr. Hoffland and the RPB team for hosting an amazing event today. I'm excited to get started. I'm especially thrilled that we are joined by two world-renowned experts that I'll introduce here in about a minute. Our event today is on Sjogren's, a chronic autoimmune disorder that is known for two of its most common symptoms, dry eye and dry mouth. Today, we'll specifically focus on Sjogren's and its effects on the eye, to specifically ocular surface disease or dry eye. This topic is especially important because dry eye is its main clinical manifestation. And we know specifically in primary Sjogren's that the presence of specific antibodies and cell infiltration in the glands changes the way that tears are produced, and it effectively reduces tear production. These changes can break down corneal homeostasis and affect the tear film and cause ocular surface inflammation, damage, and neurosensory abnormalities. These ophthalmic manifestations for patients may lead to eye fatigue, blurring, challenging vision throughout the day, and especially discomfort, potentially pain, stinging. These symptoms carry a significant burden for patients and can affect their quality of life, daily functioning, productivity, and emotional well-being. So it's a really important topic, and we're thrilled to be having these two experts walk us through some important research that's being conducted. For Dompe specifically, we've been committed to Sjogren's research over the last few years. Dompe is specifically dedicated to conditions that have high unmet medical need. We received our first ever approval from the FDA for a topical biologic for a condition called neurotrophic keratitis. But over the last few years, we've completed additional clinical trials within dry eye disease and Sjogren's dry eye disease. We completed a phase 2 clinical trial in moderate to severe dry eye patients, and we moved on to completing 2 phase 3 trials specifically within Sjogren's dry eye disease. We'll see that data later this year. We are excited to be able to share it with the organization. Our expertise is deeply embedded in various applications of neurotrophins. That includes BDNF and NGF, brain-derived neurotrophic factor, and nerve growth factor. Based on the evolving literature in dry eye, reduced tear production leads to inflammation and peripheral nerve damage. We know that the corneal nerves contribute to reflex tear production and blinking, and they regulate the epithelium proliferation and differentiation. So impaired nerve function may evoke dryness sensation, potentially pain in some scenarios, reduce epithelial recovery, and in chronic scenarios, a neurotrophic cornea. Therefore, knowing that the corneal nerve plays a critical role in the pathophysiology of dry eye disease, which has also been seen in the evolved definition by Dews too, including the neurosensory abnormalities within the definition of dry eye disease. With this knowledge, Dompe has dedicated its resources to potentially expanding the indication of nerve growth factor and endogenous protein involved in the differentiation and maintenance of neurons through very specific high affinity and low affinity receptors in the anterior segment, which may support corneal innervation and integrity. So we're looking forward to continuing to evolving our research and dedicating resources within the Sjogren's dry eye disease space, and look forward to presenting some clinical data with you later this year. So without further ado, I'm pleased to introduce our first speaker, Dr. Essen Ekpek, who I've had the pleasure of working with over the last few years and is really a world-renowned expert within Sjogren's. And I want to quickly walk you through her bio. So she is a professor of ophthalmology and rheumatology at the Wilmer Eye Center at the Johns Hopkins University School of Medicine. Dr. Ekpek has several appointments, and she's currently serving as the Director of Ocular Surface Disease and Dry Eye at Wilmer, the Associate Director at the Sjogren Center at Johns Hopkins Bayview Medical Center, and she is a member of the Clinical Trial Consortium for Sjogren Syndrome Foundation, a Medical and Scientific Advisory Board member for TFOS, and she is the president of the Foster Ocular Immunology Society. Dr. Ekpek is involved in a variety of dry eye initiatives, has authored and co-authored various book chapters on dry eye, and has led several clinical trials. Dr. Ekpek's current research interests are around dry eye disease and the impact on visual function. Without further ado, Dr. Ekpek, I'll pass it over to you so you can kick off your presentation related to ocular surface disease. Thank you so much for being here with us today.
Thank you, Georgia, and thank you, RPB, for this very important event. All right, so I wanted to start talking about dry eye first, because why? Because dry eye is really important. Dry eye affects very many people. It's estimated that in the United States alone, approximately 25 to 30 million people have dry eye. They suffer from dry eye. And the implication is that not only dry eye patients have discomfort, burning, pain, itching, stinging, but also they complain about visual fatigue, blurred vision, or fluctuating vision. In fact, a lot of the patients are misdiagnosed with various other problems in clinic because they don't know, neither patients nor doctors realize the importance of dry eye in vision. I was on the definition and classification committee, as well as steering committee of Tear Film and Ocular Society Dry Eye Workshop 2. I really like this definition because it's very comprehensive. It includes everything that has to do with the importance of dry eye. Basically, dry eye was defined as loss of homeostasis of the tear film and accompanied by ocular symptoms. Hyperosmolarity, inflammation, and neurosensory abnormalities were included as etiological factors, which is important. Inflammation is obviously, it has a big role in the development and perpetuation of dry eye, not just topical or local inflammation, but also systemic inflammation. A while ago, we looked at our patients here at the Wilmer Eye Institute with dry eye to see the association between dry eye and systemic inflammatory diseases. We found that 47.4% of patients came in with a known systemic inflammatory disease. And this included Sjögren syndrome, autoimmune thyroid disease, rheumatoid arthritis, sarcoidosis, etc., a number of different kinds of diseases. Ophthalmologists on this call might remember that uveitis and scleritis are well known to be inflammatory diseases of eye that are associated with systemic inflammation. But dry eye is not well known in that field. I propose that dry eye is same as uveitis and scleritis, except it just affects the surface of the eye. It is an autoimmune disease. Importantly, of the 81 patients of 228, 25 of the 81 patients who had no previous history of any autoimmune disease, 25 additional diagnoses were made. And most of them were occult thyroid disease, but also 5 of them were Sjögren syndrome. Sjögren's is a common autoimmune disease. It is the most common in females. Dr. Henrik Sjogren defined it in 1933. At the time, he was a ophthalmology resident at Karolinska Institute in Sweden. And he noted these 16 women with severe dry eye, filamentary keratitis, and other autoimmune systemic manifestations such as dry mouth, joint pains, and fatigue. He was doing his thesis in order to be able to graduate from ophthalmology residency, and this is his contribution. He defined the disease as a ophthalmology resident. As Georgia went over, it is a progressive chronic multi-system autoimmune disease. It can affect any organ or system in the body. Most common manifestations are dry eye, dry mouth, fatigue, and joint pains, but it can basically involve any system or organ in the body. For some reason, Sjögren's is very underdiagnosed and underappreciated within ophthalmology. Even within rheumatology it is, but especially within ophthalmology practices or optometry practices. My good friend, Dr. Vatini Bunia, who is a professor at Shea Eye Institute in Philadelphia, did this survey study a while ago and published it. One of the reasons for why Sjögren's is so underappreciated is that we as eye care providers think that all Sjögren's patients are identified and diagnosed, and there are only a few in my practice. We all expect these patients to come in with diagnosis, for example, from rheumatology or a primary care physician's office for treatment. We don't realize we're at a place to diagnose these patients. And in order to do that, obviously, we have to work up these patients. Again, a little while ago, we published this paper, which is the first in the field, demonstrating that approximately 10% of patients with Sjögren's, 10% of patients with dry eye have underlying Sjögren's. Another 10-11% have rheumatoid arthritis, but the distinct difference between the two is that 96% of the times, rheumatoid arthritis have already been diagnosed. They carry the diagnosis on presentation, but only a third of the patients with Sjögren's carry the diagnosis when they come in to see us. So we are at a place to work up these dry eye patients and identify Sjögren's patients appropriately. 2/3 of the time, the traditional antibodies SSA or SSB are positive, but another third of the patients might require a minor salivary gland or lip biopsy. So if you have high suspicion, the patient might need a biopsy. We thought that the 10% was very high, and I was a consultant to Pfizer at the time, and they were running a study trying a different medication for dry eye, tofacitinib. And I tagged along this study, adding the traditional antibodies to the workup, and we found the same 11% in the same study involving 327 patients in a prospective study. And again, This is a table from the DREAM study, the largest dry eye study that was supported by NIH or NEI. Again, it shows the same rate. 10% of all these dry eye patients have an underlying Sjogren's. Only a third come in with the diagnosis. But look, if you look at the 2016 Sjogren's Foundation patient survey, only 5% of the patients are being referred by an eye doctor for a workup to identify underlying or to assess, identify or assess underlying Sjogren's, versus even self-referral is 8%. I think this is very important. As eye care providers, we really need to look forward for these patients. Another problem with Sjogren's is that it is, and again, this is the same thing. It is aligned with Vatina Bunya's publication that Sjogren's is very underappreciated. There might be reasons for, from the patient side for why that might be happening. One important thing is that Sjogren's dry eye is a severe dry eye, but yet patients don't always complain of pain or dryness symptoms. They mostly complain of being unable to see or visual related symptoms. They don't really talk about discomfort, and that might be, in fact, because of a neurotrophic state of the cornea. These patients, after long-standing chronic, pretty significant inflammation, they develop reduction in the density of the subbasal nerve plexus in the cornea, and that might be the reason for why they have this perpetual inflammation and breakdown of the ocular surface and poor vision, fluctuating vision, but yet they don't really talk about pain, which might be attributable to the neurotrophic state. So Sjogren's related dry eye is actually really bothersome. It is more bothersome than the systemic manifestations of the disease. According to the 2016 Sjogren's Foundation patient survey. In fact, Dr. Simon Bowman in England also ran a study which did show that fatigue, systemic fatigue and ocular fatigue, probably because of the corneal findings, are the most important symptoms of Sjogren's and limiting the patient's quality of life very seriously. But when you look at the EULAR, Sjogren's patient reported index, which is ESPRI, EULAR is like our American College of Rheumatology. It's the European counterpart of ACR. And this is the most commonly used tool to evaluate patient symptoms. As you see here, maximum imaginable dryness, no dryness. There are no like vision related questions. There are no quality of related questions or anything like that, which I think needs to change. Another problem with Sjogren's is that a lot of the times Sjogren's syndrome is not seen as a significant issue. It's seen as a nuisance. It's seen as a bunch of women, middle-aged women, just constantly complaining of their eye problems when they have 20/20 vision, even though they have a lot of visual complaints. So a while ago, again, we published several papers in the field showing that Sjogren's can lead to extraglandular ocular manifestations. In fact, we reviewed 163 consecutive patient charts evaluated at our Johns Hopkins Sjogren's Center. We noted that 98% of these patients had a history of dry eye for an average of a decade prior to diagnosis. So they had this dry eye and they were never diagnosed, and by the time they got diagnosed, it had been a decade. When we looked at the extraglandular ocular findings, we found that 25% of these patients had some form of extraglandular ocular finding. in the form of corneal findings like ulcerations, perforations, conjunctival findings like cicatrizing conjunctivitis, chemosis, uveitis, retinitis, optic neuritis, etc. And a lot of the patients, 28%, between 28% to 40% had extraglandular systemic findings. And in this study, we found that 2.8% of the patients had lymphoma, a B-cell lymphoma. But if you look more carefully and over a longer period of time, the lymphoma risk increases to 5% over 8 years. Importantly, these patients that had extraglandular ocular findings were almost 4 times more likely to have extraglandular systemic findings with potential mortality, morbidity, and even mortality. And dry eye precedes these findings on average by a decade. So it is important for us eye care providers to look for Sjogren's amongst the dry eye patients. Subsequently, we also reviewed again our patient cohort and demonstrated that men patients were even more likely to have extraglandular findings, concurrent malignancies, etc. But this is important because usually men patients with dry eye are almost never suspected of potentially having Sjogren's, and they are not worked up. It is important to realize that approximately 10–11% of patients are men, and they actually have a more severe disease. Extraglandular ocular findings have been described by others as well, not in large series, but in small case series. From scleritis to uveitis to retinitis, optic neuritis, etc. But yet, when you look at the EULAR's disease activity index, which is SDI, there's only lacrimal gland swelling. And I, as an ophthalmologist who has been, in this field for about 20 years, have never seen a patient with lacrimal gland swelling. So I think ocular findings are really less well known even amongst other disciplines like rheumatology. So we as eye care providers need to be more representative in the field, because great majority of the patients, 98% of the patients, suffer from the dry eye. These are some pictures showing conjunctival findings from follicular appearance to subepithelial fibrosis, lymphoma, conjunctival chalasis, chemosis. Peripheral corneal findings could be in the form of some subtle little haze in the inferior portion, or guttering, or sometimes neovascularization, where the tear film is bathing the bottom meniscus, is bathing the cornea. There are usually these changes happening. This is a patient, actually a female rheumatologist patient, who was sent to me for pterygium surgery. But look, it is not a pterygium because it's not wing-shaped. It should have been triangular. It's not. And corneal optical coherence tomography demonstrated the thinning underneath here, and ocular surface staining was significant. And subsequently, this rheumatologist patient was diagnosed with Sjogren's by an ophthalmologist. And this is another man patient who came in with perforation of the cornea. You can see the leak. Patient did have rosacea, or rosacea is very common and it can cause corneal or ocular surface problems, but nobody bothered to stain his conjunctiva. And I did, and I finally diagnosed him with Sjogren syndrome, obviously in addition to his obvious rosacea. But I think the Sjogren syndrome or Sjogren's was his main problem. Here's another man patient, came in with this necrotizing scleritis. Workup was negative. Usually when you send a patient to a rheumatologist for scleritis workup, they get a vasculitis workup, which is ANCA, rheumatoid factor, and things like that. Like I mentioned, great majority of the time, those patients have already been diagnosed by a rheumatologist. Sjogren's is the one, along with sarcoidosis. Sarcoidosis is about the same. Ophthalmologists or eye care providers are at a place where we can diagnose these patients. Anyways, the patient was patched up, was placed on the CD20 inhibitors systemically, and did really well. Again,
Dr.
when patients.
Ekpeck, you're at the 18-minute mark. Could you wrap up in the next?
Thank you.
Please?
Thank you. I will wrap up. So again, men patients are at a higher risk, actually. They don't comprise most of the patients, but they comprise the more severe patient group. So we need to be on the look. In conclusion, Sjogren's is a very common disease, actually. It is under-recognized, underappreciated, and undercared in eye care clinics. Sjogren's related dry eye decreases the quality of life very significantly. It isn't just the discomfort problems, but the visual problems are even more important, fluctuating and blurred vision. Extraglandular ocular manifestations may be vision threatening. I have the impression, and the work is in the way, that ocular surface inflammation coupled with the neurotrophic keratitis might play a huge role in these corneal complications such as perforation and non-healing ulcers. And I would like to stop here and turn it back to Georgia. Thank you very much for the opportunity.
Thank you so much, Dr. Ekpeck. Every time you do a presentation, I feel like I learn something new and extremely important. You mentioned such important topics around diagnosis and really the patients. The patients are really struggling, and getting that diagnosis sooner makes a difference in their quality of life and vision. So really appreciate your studies that you shared, and excited to continue our journey through some of this research and knowledge by quickly introducing Dr. Cynthia DePaiva. I'm very thrilled to have you on the panel today. Dr. DePaiva is an associate professor of ophthalmology at Baylor College of Medicine in Houston, Texas. She recently received the endowed Carolyn Ellis Professorship. She was elected as the co-chair of the Gordon Cornea Research Conference and chair of the 2028 Gordon Research Conference. She sits on the editorial board of several journals and is a standing member of the National Institute of Health. She is the editor-in-chief of the Ocular Surface, a world-renowned journal. Her research interests are within the dry eye space, ocular surface disease, animal models for Sjogren syndrome, the microbiome, and aging. She has extensively published about the pathogenesis and pathophysiology of dry eye and Sjogren syndrome. Excited to pass it over to you, Dr. DePaiva, to go through your presentation today. Thank you for joining us.
Thank you for the foundation for this opportunity to be here and spend some time with you, telling a little bit about the research that I do. As a background, I've been studying Sjogren syndrome for many years, more than I care to say because I would tell my age. But I want to go on and tell more about what we've been finding. These are my disclosures. There is a patent in the United States about some of the work that I'll be talking today, and I have some consulting agreements that are not related to this talk. Since I'm coming after Hassan, I didn't spend much time reintroducing the disease, but I do want to introduce the bacteria in some terms that we use. So when we say microbiome refers to the collective name of the bacteria that live inside your body. You could also think there is a virome for the virus that live with you, and also the fungosome, which is the collection of the fungi. When I started this work about 12 years ago, we all think about bacteria as being bad because bacteria, as is shown on the picture, can cause infections. And most of the time use antibiotics to kill the bacteria. But actually, when we think about bacteria, a lot of bacteria live in symbiosis with us, meaning they live well and they don't cause any disease at all. Data from the Human Microbiome Project actually show that the human body has trillions of bacteria, and virtually any surface in our body, including the nose, the mouth, the skin, the gastrointestinal tract, and the bladder, the vagina, everything that we have, any surface that we have has bacteria inside it. And data from these studies show that we're maybe only 10% humans. So that is when you compare the number of genes that are provided by the bacteria and the number of genes that we have in our DNA. And so with that, we thought, can we investigate the bacteria that live in our eye? So the rationale for that is because the eye is an exposed mucosa, so you can touch your eye, it's in direct contact with the environment. And as I mentioned before, any body parts that we have bacteria. However, it has been shown since in the 1970s that when you try to culture bacteria from the eye, the cultures come back more than 50% of the times negative. But if we just move a little bit to the eyelid portion, so a little bit more of the external part of the eye, close to the nose and outside, then we start to have a bacteria that is positive. So we then decided to do a study using next generation sequencing. And this is a very busy slide, but what it means is instead of trying to culture bacteria, we opt to swab the inside of the eye using a cotton swab and then investigate the bacteria by the RNA sequencing that. And this study was published, and I will go a little bit quick over that, but what we found is that bacteria on the eye, it's very low biomass. I mean, there are not a lot of bacteria that lives in your eye, which is very surprising because even a normal skin has a lot more bacteria than inside the eye. We had some technical challenges because when we have very few DNA to go with, it's more difficult to identify. And even some of the DNA kits that we buy, they seem to be contaminated, meaning they already come with the DNA inside. So sometimes it's very difficult to differentiate from the eye sample and from the kit sample. But in that same study, what we found is different than the ocular microbiome, the bacteria that live inside our gut. So the bacteria that we can recover in the stools, or as I'm going to mention from now on, the gut microbiome, they are very different between Sjogren's and dry eye and normal subjects. So here in pink, we have the Sjogren's patients, and in green, we have the normal subjects. And it's almost like just by looking at the composition of the gut microbiome, we can draw a line and we can separate the two groups just by looking at the composition of the gut microbes. So the microbes are living inside us. When we did, again, 16S deep sequencing, we observed that there was difference on certain bacteria that we call commensal bacteria. They are decreasing in the Sjogren syndrome patients, which are in the black bars. But also we saw an increase on certain other bacteria that we call potential pathogens. And we call these dysbiosis. Dysbiosis means is an imbalance of the gut. It could mean more, it could be less. But one way to think about intestinal dysbiosis is not just a decrease in the numbers, but it's also a decrease in the variety. So you could see on this fruit basket here, we have a lot of fruits and we have a lot of different types of fruits. While in this other one, we have a decreased number and we have a decreased variety. So if you substitute the fruits by bacteria, you have an idea what is happening in Sjogren syndrome patients. And this was important to us because using the same LR symptom score and severity score that Essen just mentioned before, we observed an inverse relationship. So the greatest severity of the patients was found when they had very few bacterial OTUs in the gut. So this tells us that is clinically relevant for the patient. So the conclusions for this small part is that the ocular conjunctiva is not a sterile site as we thought it was, but we have some bacteria, not a lot of bacteria, but some, and there is no difference in the Sjogren syndrome patients. But there is a difference in the gut microbiome of the Sjogren's patients. What we don't know yet if it is a cause or consequence. Other studies have also shown dysbiosis and alterations on the gut microbiome. So I'm just showing you as a flavor some other studies from other groups from all over the world. They have confirmed our findings as well. So going from the human research, we said, can we try to answer the question, are these commensal bacteria, are they good or are they bad for the ocular health? And one way we can study this is in animals. Some of the techniques we can use to study are to induce dysbiosis by giving oral antibiotics, or we can do fecal material transplant, or we can use germ-free animals. And there are others also on the screen. So I'm just going to show you some of the vignettes of some class studies that we did in small laboratory animals trying to answer the question, are the bacteria good or bad for the eye? In this study, we induce dysbiosis by giving the mice a cocktail of oral antibiotics, and it's in the black bars. And we subject these mice to desiccation stress and compare it to mice that had the regular standard desiccation stress. As you can see on the black bars, the animals that don't have the gut microbes, they had worse dry eye disease. So that was the first clue that the gut microbes are protective to the eye. In this other study, we did a similar, but we did a reverse experiment. Using global cell density, we induced dry eye disease with the antibiotics, which it's down here, ABX7 for 7 days. And then we fed the mice either a PBS or fecal gavage. So this is the reconstitution experiment where bacteria are given back to the mouse. And as we can see in the graph, by doing the fecal gavage in mice that had antibiotic, these mice can recover the number of Goblet cells in the conjunctiva. We also use some germ-free mice. So germ-free mice are mice that are born via C-section, and they're born and raised in small bubbles like that, where everything that comes in contact with them, including food, water, and touch, is all sterile, either autoclave or irradiated. So these mice never have any type of bacteria. These mice are relatively normal, but they have huge alterations on the immune system, because the immune system is how we relate to the environment. And so one way to think is germ-free mice don't have any bacteria, not just in the gut, but all over themselves. And when we investigated again, conjunctiva goblet cell density in these mice, which are shown here in these purple magenta cells, we observed that germ-free mice have a decrease in the number of goblet cells. And if we do a fecal gavage, just like the experiment before, we can actually reverse the dry eye phenotype. Goblet cell loss is a frequent finding in Sjogren's and in dry eye patients. And this was very important. So the conclusions that we want to say is that there is a gut-eye lacrimal gland microbiome axis where the microbes are protective to the ocular surface. And not just the eye, but there is a great influence showing that the gut microbes can also influence the brain, the liver, the bones, the kidney. So the more we learn about these microbes, it becomes more obvious that it's important to keep them and keep a good gut microbiome. And in my last part, I just want to show a little bit about short-chain fatty acids, which are produced by certain members of the gut microbiome. And they are mostly well-known are butyrate, propionate, and acetate. There are others, but I'm just actually going to be talking a little bit briefly about butyrate. So very surprisingly, we found that the cornea and the conjunctiva, they have a specific butyrate transporter that has a very long name, but it stands for SLC58. And staining in dark red, you can see that there is a staining in the cornea and also in the conjunctiva. So that means the eye has a possibility to respond to this metabolite that is produced in the gut. And when we did a study where we fed the mice tributylene, which is a prodrug of butyrate, it smells of margarine. We, during the same desiccation stress model I mentioned before, we can completely blunt the dry eye disease that we can induce in these mice. So this was a very impressive because this was not an eye drop treatment. This was only fed. So just oral administration was enough to change the dry eye and promote ocular health. And we also found that this treatment is to decrease in type 1 interferon genes. And we think that is important because Sjogren syndrome patients have what we call interferon signature that we also reported in the conjunctiva. So it's in the blood, it's in the eye, and it's also in the conjunctiva. Showing on the screen are some Sjogren's patients. There are these over here, and a lot of healthy control, and this is investigating gene expression. So the conclusions that very surprisingly, the ocular surface epithelium express the transporter for these fatty acids, butyrate. You can give these to mice orally, and it can improve the dry eye signs, and it does so by the type 1 interferon gene. So if you would put it together, we know that the bacteria in the gut are producing metabolites. Some of these metabolites are acting through the dendritic cells or other immune cells, such as regulatory T cells, and either indirectly through these cells are going back to the eye to conquer and to offer protection, or maybe even to a direct effect to the plasma and to inhibition of other inflammatory mediators. So the take-home message for you is the microbiome has a protective role in animal models of Sjogren syndrome, and it's maybe possible that this effect is directly by the bacteria or maybe by the products. Before I wrap it up, I'd like to acknowledge people that have done a lot of the work. These are the people in the germ-free facility, is very high specialized. Some of my collaborators, including Rob Britton and Laura Schaefer in the Department of Microbiology and Virology here at Baylor College of Medicine, but also Stephen Fulfeld and Deacon Lee, which are my former mentors, which I work closely, and other members of my team that have been working together with me in this project, but also the funding. In regards to research to prevent blindness, in collaboration with Rob Britton before, we had a Stein Innovation Award specifically to look at the role of the microbiome in dry eye. So we're very thankful to the foundation. And with that, I can wrap it up and answer any questions. Thank you.
Thank you so much, Dr. DePaiva. That was such an interesting talk, and microbiome is such a really interesting topic as it relates to Sjogren. So thank you for walking us through some of your fascinating research. I'm excited now for the opportunity to open it up to questions. Thank you for using the Q&A component that was down at the bottom of your screen. So I'm going to start with those questions, and I'm going to kind of pass it back and forth between Dr. Ekpeck and Dr. DePaiva as the questions go to different areas within your talks or in general. So let's kick it off with the first question that came in. Do patients display any retinal abnormalities? Maybe Dr. Ekpeck, we can start with you and you can kind of walk us through some of those clinical pearls. And then Dr. DePaiva, please feel free to also give your contribution as it relates to, do these patients specifically show any clinical manifestations related to the retina?
Yes, indeed. It is not the most common manifestation, but a lot of the patients with Sjogren's have retinitis. In fact, some of the time, and it's not a very obvious inflammation because they don't get vitritis, no cells or flare in the anterior chamber either. So a lot of the times we as eye care providers get confused between Sjogren's related retinitis versus retinopathy from the treatment that is used for Sjogren's, like Plaquenil or hydroxychloroquine. Yes, it does happen, and one needs to be very careful to know the association.
Absolutely. Anything to add to that?
No.
No, that's great. I think this is a very important question and a little bit broader, maybe not necessarily related to your clinical practices directly, but if someone is diagnosed with Sjogren's, how do they locate the best ophthalmologist to see? How do they find out about current clinical trials, potentially to participate themselves?
Yeah, this is a great question. Let me tackle it first. I'm going to take the first stab. So basically, our Sjogren's patients are well educated. They're thoughtful. They look out for themselves. They advocate for themselves. 8% of patients were referred by themselves versus 5% from ophthalmology for a workup. Yes, they can always contact Sjogren's Foundation. Sjogren's Foundation maintains a database for current and upcoming trials, and they can make suggestions for providers.
Yeah, absolutely. That's a fantastic website. If you haven't gone to it, I can also drop the link into the chat feature. It's broken down by state, so you can actually go down to even your zip code, what's the closest within your area. They also have a newsletter that you can sign up for. So anytime something new comes up in your area, they'll also alert you via email, which is, I think, a really new, exciting feature that Sjogren's Foundation has put together about 1.5 years ago. I think both of your centers are probably world-renowned for treating Sjogren's. And I think that if you can't find somebody locally, traveling to one of the bigger centers that sees a lot of these patients, if you haven't been able to get the proper diagnosis, is always a good way to start if you can't find somebody within your local area. Can you explain the second round of conclusions again about the short chain fatty acids? So Dr. DePaiva, I'll pass this back to you. The second round of conclusions related to the short chain fatty acid slide.
Sure. So short chain fatty acids are produced in the gut by certain bacteria after they fermented the food. So it comes from foods that are very rich in fiber. So anything that has a lot of fiber, think beans and green veggies on that. Once they are in the gut, they are be fermented in certain bacterial communities, meaning certain types of bacteria, not all of them, will produce these metabolites that can then enter the bloodstream and can reach the plasma, and then it can go to all over the body. So the eye is obviously very separated from the gut. There is not a directly communication. But what we found was the cornea has a transporter that can take the short-chain fatty acids from the plasma into the cell. So without that transported, even though if you have it available, you'll not enter the cell. So you need a transporter for that. And we described that there is the cornea and the conjunctiva have, meaning if there is short-chain fatty acids in the plasma or in tears or in other fluids, it could enter the cell. And as a proof of principle study, we fed mice with tributylene, which is it's not the same drug. It has a glycerol and 3 molecules of butyrate. It smells better and it's easier to handle. So we use that as a proof of principle, and we fed mice these during our inducible dry eye disease, and we found that it can blunt. In other words, it can improve the cornea staining in these mice. So the conclusion is, surprisingly, the cornea is capable of responding to a metabolite that is made in the gut. And if you administer that during dry eye disease, it can actually improve the dry eye.
Very fascinating. That's a topic that I've always been super intrigued with. So one additional question that came in, actually we have 2 additional questions, and I know we have about 7 minutes to get through them. So let's start with, what are the biggest challenges you identify in breakthroughs for Sjogren's? What are the biggest challenges you identify in breakthroughs? I'm going to guess that maybe kind of the rebound effects of the disease, breakthrough maybe through therapy. I don't want to make a conclusion about what breakthrough specifically for Sjogren's, but what are the biggest challenges you identify in breakthroughs for Sjogren's?
Well, I'm thinking that, well, I don't know if this answers the question, but I have something on my mind I wanted to say, and that's that a lot of the clinical trials, if they fail because the trials include patients who have had chronic disease. The late diagnosis is an issue. So underappreciation and underdiagnosis and late diagnosis are related. So then by the time the patient has a diagnosis and seeks treatment, the inflammation is too chronic and the extent of damage is too significant. So I think that that's one of the challenges in the management of Sjogren's.
Yeah, I would totally agree. If I may jump in, I think diagnosing Sjogren's, and because we still rely on a biopsy positive to diagnose Sjogren's, a lot of times waiting the patient to get severe enough to be diagnosed, and then we know when we have a biopsy that is positive, there is a lot of damage that already happened, and it may be irreversible damage. So I think diagnosing early and being able to treat early, it remains a key issue in Sjogren's,
Absolutely. And you make a really great point around the clinical trials. It's this kind of double-edged sword where you're almost diagnosing them at the point of enrollment, and then at that point they're too severe and they don't qualify for the trial. So it's absolutely a challenging environment for these patients. If we didn't answer your question appropriately, please type it back into the chat, and we'll make sure to address it. And one final question in our last couple minutes together here, Dr. Ekpek, was for you. How do we overcome the gender biases in diagnosing and treating Sjogren's that you highlighted specifically in your presentation?
Yeah, that it is very important. Yes, for some reason we don't ever suspect of Sjogren's being the underlying reason for men patients with dry eye. I think that, much education is needed in the field. Sjogren's is known as middle-aged women's disease, just nuisance, bunch of complaining postmenopausal women. So very, very rarely men patients are tested for Sjogren's. Another problem with men patients is that they tend to be triple antibody negative. So they, more often than women, they require a minor salivary gland biopsy. I think it's education. I think that's very important. I just want to say one last thing. I think tear film could be very important for diagnosing Sjogren's. We have shown that there are differences between Sjogren's versus non-Sjogren's dry eye when it comes to the tear film cytokines and chemokines. I know that Dr. Sharmila Mazli in Boston is working on this field for early diagnosis, maybe with even more collaborations with Cynthia and clinicians. This could be a good field to investigate, I think.
I have a question I'd like to ask, if I may. You were talking about the difficulty of proper diagnosis and relying on biopsies. I'm wondering if there is somebody who is looking at this using artificial intelligence tools. So if one could develop an algorithm to have better diagnostic power, that could be a great advance for the field. I don't really know if anybody's looking at this or not.
I don't know of anyone specifically looking at Sjogren's, but along with Dr. Pradeep Ramalu, who is the head of glaucoma at Johns Hopkins Wilmer, we are looking at dry eye using artificial intelligence to see if, based on patient symptoms, one can differentiate dry eye versus blepharitis versus glaucoma versus cataract, other things. But I think you have a great point, Brian, that AI could be very useful because we can screen a lot of patients within a very short period of time.
I wonder if the IRIS data set could be used, which has a large number of patients. I mean, I don't know, how often Sjogren's would show up, but that could be an interesting data set to possibly look at to see if to begin to at least generate some hypotheses that could be useful.
I couldn't agree more. Maybe we should apply for for a grant support from IRIS. Yes.
Right, and IRIS is a large data set that's run by the American Academy of Ophthalmology, but it's larger actually than the Medicare data set. So I will draw us to a close. We're nearing the end of our time, but I want to thank Dr. Georgia Posadas for opening the session and very ably moderating it. And I want to thank you, Dr. Akpeck, for just your wealth of knowledge and for really giving us a great overview of Sjogren's disease and drilling down in a lot of different areas. And then finally, Dr. Cynthia DePaiva, for really taking us into the ocular biome and kind of taking us deep into this really interesting connection between the biome and eye disease, which I think, even 5 years ago or 10 years ago was just totally unappreciated and even kind of like ridiculed. So you're a real pioneer, and I appreciate the work that you're doing. I think your research opens up some possibilities for possible treatment in the future. So very exciting. Again, thank you to everyone. A very rich, and I think this is a somewhat underappreciated area, and there's not enough attention given to it. So thank you to all of you for drawing attention to it, and I think raising our level of knowledge. Well done. Thank you.